Hello and welcome to Agri-History, Tropical Edition. In this episode, we'll cover the domestication and origins of sugarcane. Multiple species of sugarcane are used in sugarcane production. Three predominant species are commonly used. These species include Saccharum officinalis, which is the most common form of sugarcane cultivated and domesticated. Today's cultivars of sugarcane are hybrids made using the species. The other species is called Saccharum sinensis, which itself is a hybrid between Saccharum officinalis and Saccharum spontaneum. This species was domesticated in the Papua New Guinea area and further improved by the Chinese. And the third major species would be Saccharum barberi, which was domesticated shortly after the introduction of Saccharum officinalis in India. These three species will respectively be called S1, S2, and S3 for brevity's sake. Sugarcanes, generally speaking, are large perennial tropical/subtropical grasses that are harvested for their sweet liquid, which is used to make sugar. In terms of tonnage produced of sugarcane is number one. In terms of how many calories are consumed that are made of sugarcane, it ranks number 3. Now on to the origins of sugarcane, specifically the S1 species. S1 has a natural range all across Southeast Asia, which includes the Malaysian archipelago, New Guinea, and India, as well as several island groups in the Melanesia area. Current evidence, both archaeological and genetic, indicates that the S1 species had its origins in the New Guinea region and was probably domesticated from the wild species Saccharum robustum. There's also been research going on to see if any other species in that area contributed to its genome, and it was shown that our S1 cultivars, which are also known as noble cultivars of sugarcane, are actually complex hybrids of S1, S2, S3, as well as a few wild species like Saccharum spontaneum and Saccharum robustum. But I digress. It is likely that the S1 species, after being domesticated, spread to the other regions of the world, and it's likely that this species became a parent species for the S2 and S3 species, which we'll cover next. The sugarcanes commonly grown in North India and China are the S2 and S3 species. Based on current genetic evidence, which we'll cover a little bit later on, it is likely that both species were derived from crossbreeding between S1 and Saccharum spontaneum. This likely will have happened around 1000 BCE. As of right now, the S2 and S3 species are only used in collections and breeding programs. The first hypothesis of these origins are the S1 species independently crossbred with Saccharum spontaneum in two different areas, China and India, and these independent hybridizations would become their own species in time. The genetic evidence shows that between 61% to 66% of the DNA in the S2 and S3 species comes from the S1 species, and the rest comes from Saccharum spontaneum, and this remains constant for both species. So it's likely that there is one initial center of origin for these species, and once the hybridization is finished, geographic separation and mutations that happen over time would cause these populations to diverge from each other and become the two species we know today. Now on to the breeding history of sugarcane. Now on to the history of sugarcane breeding. Sugarcane was cultivated and bred through open pollination throughout most of history. And as sugarcane moved from country to country, it crossbred with native species, oftentimes creating new variations, as well as the S2 and S3 species. The quote-unquote noble forms of sugarcane discovered by Linnaeus were collected, and the most common type grown in the New World was the Indian cultivar Creole. This replaced the previous cultivar Otahyte in 1793. The S1 cultivars, or the officialis cultivars from the Pacific Islands, replaced the less productive Indian varieties in the New World, and for over a century they were the major source of sugar in the New World. Unfortunately, these noble varieties were also susceptible to various diseases, and those were later replaced by new hybrids from emerging sugarcane breeding programs. The first breeding program started in Java and Barbados in 1888, after the discovery that sugarcane can produce viable seed. And as mentioned before, these days, most cultivars of sugarcane are complex hybrids between many different species to get what the people call noble traits, which is to say the traits of the S1 species. They often do interspecific hybridization, i.e. crossbreeding between two different species and then back crossing into the S1 species, until only the resistance traits remain. This process started in the early 1900s. Afterwards, from the early 1930s onwards, most hybrids are crossbreeds between nobilized canes, which is to say, the results of all that backcrossing. So the future hybrids that were bred for sugarcane production were all developed from those cultivars of S1, with a little bit of DNA from a wild parent integrated into it. Well, that covers everything. Thank you for watching, and stay tuned for the two-part final for Agri-History Tropical Edition. Stay lit, fam.